Hey everybody, my name is Joseph. I'm your learning assistant. I'm sure most of you know. This video is going to be a review session for exam 2. If you look at your syllabus for exam 2, this go around is just going to be genetics, microbial growth, bacteria, and fungi. I know that your syllabi says viruses and antimicrobial substances. You did not get to that in class, which that will go on to the next exam, which will be exam 3. So for this one, you only have 4 chapters, or 4 PowerPoints to worry about, which, in my opinion, is good for you guys because bacteria and viruses are a lot of memorization. So now you're going to be able to break it up. You're going to be able to memorize bacteria, and after this, your memorization skills hopefully will be increased, and you'll be able to memorize viruses hopefully with a little less struggle than you did with bacteria. Dr. Kogash did post your study guide, I think at the beginning of this week. So you have about a week to go over it. It's pretty simple. Viruses are on there, so since it's not going to be on the exam, since you have not got to it, don't worry about that part. So it's pretty simple, page and a half. But like I say, Dr. Kogash says, and I'm sure other professors and other classes you've taken, the syllabus and the review session are not going to be the exam. So like you just saw, there were syllabus changes. So you can't always rely on the syllabus. So you have to go to class to get the information you need what's going to be on the exam. The review session, or not the review session, the study guide Dr. Kogash posted, if you did not go to class, you would not know that viruses are not going to be an exam. You would do it, which would be great for your next exam, but wouldn't be so great because you would spend time studying something that's not on this exam when you could use that time to designate towards bacteria. So I'm just going to go over the PowerPoints. I'm going to put out the big things I think you guys should look at. I took this class going on 5 semesters ago. I took 305, but I have been Dr. Kokash's LA. This is my 3rd semester doing it. So as I do not know what is on your exam, let me put that out. I've had a lot of people ask me last exam, and especially this exam, what's on the exam. I don't know. Dr. Kogash did not share that with me, nor is she able to share what's on the exam with me. So when I see the exam, it's the first time you guys see the exam. That's why when I come in, I look at it, and I'm like, oh. The way her exams are put together, though, comes from mostly the PowerPoint. You do have to read your book, so please keep up with the readings, but the PowerPoints are going to help. Of course, the study guides are there to help you move along. So even though that specific question might not be on the exam, a variation of that question will be on the exam. So I'm just going to go ahead and start. If you look at your review session, if you pull it up right now, it first starts with genetics. Genetics is a pretty difficult topic in microbiology. It's one of the most important, if you ask me. I'm sure most people who take microbiology are as a biologist would say, oh, maybe it's this or this, but to me, it is one of the hardest and one of the most important subjects in the entire class. So if you want to go ahead and pull up your review, or study guide, whatever you like to call it. The first question is, what is a genotype? And of course, a genotype is the genetic makeup of that specific organism. And then of course the phenotype, which is the actual trait that is expressed due to the genotype. I have seen that question, a variation of that question on the exam. It could be multiple choice, it could be matching, it could be A, B, it could be true, false. But I have seen variations of that question on the exam. Please know that. Please study it. And as you go through the PowerPoint, there's a bunch of main things. Of course, what is genetics? What is the genome? Of course, genotype, phenotype. Eukaryotes, prokaryotes. You guys should be experts at that by now. That was on exam 1. Please remember it, and please don't forget the information from exam 1, as your final exam is cumulative. And even though exams 1, 2, and 3 are not cumulative, they do use information you learned from previous exams. Please do not forget it. Please do not forget it. So you guys should definitely know what eukaryotes and prokaryotes are. When we're going to talk about DNA, remember 4 letters, A, T, G, and C. That's gonna be big. It's gonna be important. So just remember that. I'm giving you a hint to remember now. To talk about how DNA is pretty much, made together, there's 3 stages: replication, transcription, and translation. Those are your big important 3. Remember the steps to those. Those are very, 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 very important. So remember replication, transcription, translation, in that order. So just remember RTT, replication, transcription, translation. DNA and RNA. I know you've probably learned this from other biology classes because you needed to have 2 before you could register for this class. DNA is, of course, a double helix. RNA is a single helix. Remember that the DNA, of course, twists together. They're strands held together by hydrogen bonds. The strands are held together by hydrogen bonds. I do believe I've seen that on one of Dr. Kokash's exams. That was a question on my microbiology exam with Dr. Pillais for 305, and that was a question on my genetics exam. So remember it. It is very important. The strands are held together by hydrogen bonds. And of course, there's AT and CG. I told you those letters come up. So the hydrogen bonds go in between the strands AT and CG. A goes to T and C goes to G in DNA. And RNA, of course, the T is not an RNA. The T goes to a U, which is uracil. So the A goes to U and the G still goes to C. So like I said, remember the steps of replication. Please remember where replication begins, which of course is the origin of replication. It's the origin of where the replication process starts. Remember the specific enzymes that are required for replication. So remember DNA-dependent DNA polymerase. Say it again, DNA-dependent DNA polymerase. Remember, there is a leading and lagging strand. The leading strand is continuous, the lagging strand is not. And remember, the lagging strand has, I've never really said this, I think it's Okazaki fragments. So remember, the leading strand is continuous and the lagging strand is not. Remember, DNA ligase, they are the ones that tie together the Okazaki fragments. So you have the DNA-dependent DNA polymerase, which is the main enzyme that elongates the new strands of DNA. And then you have the DNA ligase, which ties together the Okazaki fragments in the lagging strand. Do know the direction they go in. There is a photo. It is on slide 12 of your microbial genetics PowerPoint. I would remember that. It just shows you where the leading strands go, the lagging strands go, the Okazaki fragments. So I would know PowerPoint slide 12 for microbial genetics PowerPoint. Then I would remember, of course, transcription, which is the synthesis of RNA to DNA. The enzyme used in this is DNA-dependent RNA polymerase. So for replication, it's DNA-dependent DNA polymerase. And then for transcription, it is DNA-dependent RNA polymerase. Remember where, of course, it starts at? It starts at an A site at the promoter. So it starts at the promoter. Of course, this elongates from the 5 to 3 direction. And then you have, for the transcription part, please remember your codons. Please remember, of course, the 4 nucleotides are A, U, C, G. T is not involved in transcription because you are using RNA polymerase, and RNA does not have a T, it has a U. So please remember your four nucleotides that are used would be A to U and G to C. You have one start codon, that is AUG, and you have 3 stop codons. which are UAA, UAG, and UGA. They are non-coding. They do not code for anything. They stop the process in its tracks. Remember how many you have. So if you have a total amount of 64 codons, but you have 3 that do not code for anything. They are just the stop codons, which is UGA, UAG, and UAA. So if you have 64 altogether, and you have 3 that do not code for anything, you have 61 that actually code for a specific amino acid. I would know, of course, that mutations do occur. Mutations happen all of the time. Some things that can happen, it can alter the phenotype. Of course, they're spontaneous. They can be induced. So here are the different types of mutations that you need to know for this exam. Know your point mutations, or it's called base substitution sometimes. They can be a missense, which it really doesn't. It changes an AA to be used, but it doesn't really. Nothing's happened. There's a nonsense, which is there's no changes results in a stop codon. So nonsense. There's no nonsense about it. You're stopping. So just remember, act like the nonsense mutation is like your parent getting mad at you. You're acting crazy. This is nonsense. Stop it. A nonsense promotes a stop codon, which stops you in your tracks. You have a silent, which the amino acid or protein changes, but of course it's silent. You have frame shift. Frame shift is the insertion of one or a few base pairs. So you insert, say you insert an A. It was, say, AAA. And now you've inserted another one, so now it's, AA is not really a good example. Say it was AGA. And now you inserted an A at the beginning of that, now it's AAG, which is a completely different amino acid that it codes for. So no, just frame shift. It does one or a few. There are some excellent examples, picture examples of these different mutations. They start on slide 30 of the microbial genetics. I would look at those. It's perfect examples. Like I said, I do not know Dr. Kokash's exams would be, but if I were going to make one of these exams, I would put something similar to this. I would put similar photos, and I would say, okay, what is this? It would have one strand, and there would be after strand, and it could be something different, and then you have to say what mutation just occurred. So please look at the photos. If that is not on the exam, the photos are not on the exam, it'd be perfect because it could just be letters strung out that represents a photo, but there is no photo background. It's just letters strung out, probably 30 letters in a sequence, and it's going to say what mutation happened. So know your mutations, definitely. Know that there, of course, causes there's radiation that can happen to sequences, and radiations cause thymine dimers. Thymine dimers are the ones that kind of bulge out, and they can cause a bunch of different problems. Of course, you have something that can repair thymine dimers. It's photolysis, which separates the thymine dimers, and there's nucleotide excision repair. Nucleotide excision repair. You can find an example of that on slide 38 of the microbial genetics PowerPoint. And then, of course, there's different ways that you can transfer your bacteria. If you go to slide 41, it gives you a list of the different transfers. The first one is transformation. Transformation was founded in 1928, or discovered, I might as well say. And it's just a transformation of naked DNA. So the cell comes around and it sees just a DNA stranded, and it picks it up. The way I remember it, it's like a penny. If you see a penny on heads, you're going to pick it up because it's lucky. It's just naked. It's just sitting out there in the environment. Then you have transduction, which is transfer of genetic material by viruses. You can either have one can be the virulent or lytic phage, and the second one can be a lysogenic phage. You can have generalized, of course, or specialized. And then you have, there's a bunch of examples. Then you have conjugation. There's something specific you need for conjugation to happen, and that would be a sex pilus. So the male is going to be the F+. They are considered F+. And they are the donor cell that is carrying the F plasmid. And you're going to have the recipient, or the female, which is going to be F-. Just remember what plasmids are. Plasmids are just, I think I mentioned this in my first video for exam 1. Plasmids are small extrachromosomal DNA. They have some kind of important roles, but they're not really essential to the normal growth of bacteria. And then I did get a couple questions on slide 72 of microbiogenetics on Piazza. It's pretty much what I just mentioned. Just remember the male is the F+, which is the donor cell, and F- is the female and is the recipient cell. And if an F+ crosses with an F-, you're going to get 2 F+ cells, which means F+ transfers the X factor to the F-, which then simultaneously happens at the same time and makes 2 F+ cells. You're going to have both F+ factors within both cells. So 1 F, let me say that again so you remember, 1 F plus mixes or crosses is a better word, with an F-. F+ then simultaneously happens in both. It transfers over and you get 2 F+ cells. If an F factor is incorporated, and of course, if F factor is incorporated into a cell's chromosome, it's an HFR, which is called a high frequency recombinant. And if we cross an HFR with an F-, we get an HFR plus an F-. So we're going to get one high frequency recombinant, and we're going to get a recipient or a female in terms that you do remember, whichever one helps you the most. Because usually only a portion of the F plasmid is transferred. If you need a visual of this to help better the process of learning this video, because I know it's hard, I know it's a lot of different things that happen all over the place. If you are a visual learner, please go to slide 74 of microbial genetics. I know sometimes that auditory skills do not work when you hear this and it's a bunch of mumbo jumbo, because I know that is with me. I need visual. Slide 74 is perfect. So just remember, if an HFR, which is a high frequency recombinant, is crossed with, if it is crossed with an F-, we're going to get an HFR plus F-, because only a little bit of the F plasmid is transferred. If you mix an F+ with an F-, you're going to get 2 F+. That's pretty much it for microbial genetics. I know that's a lot, but is a very important subject in the field of microbiology. So I would remember a lot, pretty much the entire PowerPoint. Like I know Dr. Kokash has said, and I've said, this is a memorization class. You're going to have to memorize. And unfortunately, it's not a class that you can memorize and then forget. Because you're going to have to know this not only for your final exam, which is cumulative, you're also going to need to remember this in your field. Even though you don't specifically think, oh, I'm going to have to remember this, you definitely will. I promise you, you will. Especially for nursing and definitely for global health majors. So it's better to learn it now, so when you have to take exams for it further in your career, you'll already know it. You only have to do a little bit of studying compared to a lot of studying if you don't study for this. So it's the way I look at it. You gotta look at the positive. You know, the glass is half full instead of half empty. Then you have the growth control. This one is pretty intense, if you ask me. You have a lot of information you have to learn. But like I said, learn it now and you'll be perfect. So first I would know the 3 ways to control microbial growth. To start right off, you have physical and chemical, which is group number 1. You have 2, which is chemotherapeutic, You have group 3, which is immune system and vaccines. So I actually received a question on Piazza, and if you give me a second to pull it up, it was, what is the difference between antiseptic, disinfection, and germicide? So the main difference I would point out is that the germicide is in group 2, which is chemotherapeutic, and the antiseptic and the disinfection is in group 1, which is the physical and chemical. That's the biggest. But to break it down, the disinfection is the Lysol you use to wipe down your tables after yourself, your children, your parents, or your friends eat at your table and they might have dropped something. You pick it up and you wipe it with Lysol wipes, or you wipe it with Clorox. That's a good disinfection. You're disinfecting. You're killing the vegetative bacteria and microbes that are on the surface, the table surface. Antiseptic is good to work against infections. So if you watch shows like Grey's Anatomy, you see them squirt iodine before they make their first incision. As a kid, when I used to get cuts and I used to play outside, instead of just banding it up and putting Neosporin on it, my mom would put iodine on it. And yes, it would dye my finger for a couple of days, but it actually really worked because it's really good against infection. And I used to live in the farms in a gravel road, so when I used to fall, I used to get gravel all inside of my hand. So it would perfectly keep infections away. And then germicide, which is in group 2, which is the chemotherapeutic, that kills microbes, but not necessarily all endospores. So you have different kinds, like bactericide, which kills bacteria. And remember germicide, which side is kill. So you got bactericide, kills bacteria. Fungicide, which kills fungus. Virocide, which kills inactive viruses. So it's just pretty much, it kills more than what a disinfectant would, but it doesn't necessarily kill endospores. So that is the major difference. Also note that in group 2 there is sterilization, which sterilization is it sterilizes, which includes bacteria, endospores, viruses. A good sterilization would be a high pressure dishwasher. I wouldn't go as far as saying the ones you see in houses, but go as far as ones in your chemical labs, ones that they might use in organic chemistry, that they use to wash the different chemicals out of a flask. So that is what I would think of. A very high power, maybe boiling. You sterilize the water. So where we live in Stevens City, we actually have a lot of chloride in our water. And even though our safety department here in Stevens City said it's safe to drink, I don't trust it. So if I'm going to cook with it, I'm going to boil, my macaroni in it, or I'm just going to use it for tea. I'm going to make sure it's boiled thoroughly. I boil it for 10 minutes at very high temps. I make sure it's bubbling over. And yes, some's evaporated, but at the end of the day, I'd rather be safe. So it sterilizes my water. Then you have group 3, which is degerming and sanitation. Sanitation, it just removes pathogens on food. So you sanitize service items. So once again, you go to a restaurant, they're going to sanitize by putting your fork in the dishwasher. They're not going to give you just wipe with their cloth and give it to you to eat. You also have degerming, which removes transient microbes from your skin. Degerming is like where they wipe with alcohol right before you get injections. I know all of us had to go and we had to get shots right before we came to college. So they wiped, of course, with an alcohol wipe, and they gave you the injection. That's what I would remember for degerming. Remember, I just mentioned side or cidal is kill. You kill the bacteria. Then you have bacteriostatic, which just inhibits the growth of the bacteria. I have seen that a lot on exams, so I would definitely remember that. Then I would remember conditions to consider. So the first one is temperature. Lower temperatures decrease the rate of microbial growth and increases at higher temperatures. For you guys that are in 306, you guys do this a lot. You will get an agar plate, you wipe the bacteria on it, and then you put it in a refrigerator that's at 38 degrees, and it grows, and you put it in a colder refrigerator so it stops. So you have time to look at it when you come back into the lab. That is what you are doing essentially in the lab, because the high temperatures allow for the bacteria to grow, and then the refrigerators you use in the cold refrigerators slows it down and stops it, so you can go back into the lab a day or so later and be able to record your results. Second is consider the type of microbe you're working with. Endospores are the most resistant. Of course, fungus spores, vegetative cells are the least resistant. That's why you're able to just wipe them off your surface with a Lysol wipe that you can get at Dollar Tree, Dollar General, or Walmart. Then you have the physical state and the number of microbes. So pretty much cells that actively grow at the log phase are more susceptible than dominant cells. Also, things to remember is your cell's environment. You know, what's surrounded by? Where is it at? What is the pH? You know, just remember the environment. Then I would remember the physical methods. So earlier you had the 3 ways to control it, which was physical, chemical for group 1. Group 2 was chemotherapeutic, and third is viruses and immune systems. These are the physical methods to control. You're going to have 7. You're going to have moist heat, dry heat, filtration, cold, desiccation, osmotic pressure, and radiation. So moist heat is of course going to be your boiling, your autoclaving, your pasteurization, and as we all should know from Louis Pasteur, what pasteurization is and what it is used for. I would just know the aspects or the characteristics of each. So boiling, of course, breaks H bonds, kills most vegetative cells, fungi, and disinfects, does not sterilize, but kills most pathogens. So remember, it does not sterilize, but it does disinfect. Autoclaving, I'm sure you have been mentioned that some of George Mason's equipment is used in an autoclave. It is not something that melts it. It is not something that destroys it. It's pretty much just moist air, and it's steam. Well, I wouldn't say air. It's steam that replaces the air, and it sterilizes the equipment used. Pasteurization, of course, we all know what that is. It's used on our milk, our cheeses, our yogurts, our beer, our wine. It kills most microbes, and of course it disinfects, but it does not sterilize. So we have boiling water and pasteurization, which disinfects and does not sterilize, and then we have autoclave, which does sterilize. Then we have moist heat. This is mainly used to sterilize milk. If you look, it's a very rapid process. If you crank up the temperature to 140 degrees Celsius, it can happen in a matter of 4 seconds. And this is what allows for you to be able to keep your milk at room temperature for a certain amount of hours. So the second one, that was moist heat, which is number 1. Number 2, dry heat. Dry heat is incineration. This is what I mentioned that autoclave is not. This is burning. This is incinerating whatever you are trying to get rid of. So just remember, dry heat, flame, incineration. Group 3 is your filtration. Filtration, the way I remember it was a cheesecloth at the bottom of a flask, inside of not a flask. The cheesecloth is within a funnel inside of a flask, and it filtrates the dirt and grime. Kind of think it as a Brita filter. You put the water at the top, and it filtrates all the particles, and you see these little black particles left, and your water is clear. That's what I remember for filtration. Next is cold or freezing. Of course, refrigeration at 0 to 7 degrees Celsius is bacteriostatic, not bactericidal. So that's what I mentioned by putting your microbes that you have on your agar plate in Bio 306 into the cold refrigerator. It does not kill it, of course, because then you'll never be able to get your results. It just stops it from growing, so you'll be able to get your results for that amount of time that it was growing inside of the refrigerator that is, I guess not the refrigerator, but the fridge that you use for Bio 306 that is at a 38 degrees Celsius temperature. Number 5 is desiccation, which is the removal of water. So the water is required for most processes that involve life. So if you use desiccation, it removes the water. So this is bactericidal. It kills it. Bacteriostatic, I'm sorry, excuse me. Replace that. It's bacteriostatic. It's not necessarily bactericidal, but it can be. It can be. Then 6 is osmotic pressure. If you remember from exam 1, the hypertonic, isotonic, this creates a hypotonic environment. So the osmotic pressure is higher outside than it is inside the microbe. So the water is leaving the microbe and is going to the outside environment. This is bacteriostatic. It is not bactericidal. And then number 7, you have radiation. You have ionizing, which is X-rays or high energy beams. And then you have non-ionizing, which is UV light. Unfortunately, you have to remember all of those. Then we have good chemicals used. This is another group you will have to remember. It's 11. There's 11 different subcategories. Phenols, chlorhexidine, halogens, alcohols, heavy metals, surface agents, quaternary ammonium compounds, organic acids, aldehydes, gaseous chemosterilizers, and oxidizing agents. For each one of these, I would know is it bacteriostatic or bactericidal? And then as that one concept Matt pointed out, know is it sterilizing? Is it a disinfectant? Is it antiseptic? So that's what I would know from those. I would definitely know those different categories. Slide 53 of the control of microbial growth PowerPoint. I know most people have skipped over it. Please do not. I do not know if it's going to be on this exam. I was on my exam for Bio 305, which is the same class, for biology majors. Please know this. It is a slide that goes from most resistant to least resistant. So the most resistant, you have the very tip top is prions. So it goes prions, endospores, microbacteria, cysts, protozoa, vegetative protozoa, gram-negative, fungi, viruses, gram-positive, and the very least resistant is viruses with lipid envelopes. Please remember slide 53. It's a very important slide. Then we go to, I'm going to skip bacteria for now because that is the most one that's going to be time consuming. So if you guys want to go to fungi, fungi, it is a very important chapter. I would know, of course, yeast and molds. Yeast are multicellular. Yeast are unicellular. Scratch what I just said. Yeast are unicellular and molds are multicellular. Know the way they sexually produce. One's sexual, one's asexual. Know which one is which. What else? Like I said, one's asexual, which would be yeast. Unicellular, asexual, yeast. Multicellular, sexual, molds. I'm sure she went over mycelium. I would know mycelium. There's different asexual spore types. There's different sexual spore types. I would definitely know those. But for this, I would pretty much just know what is superficial, what's opportunistic, cutaneous. But a lot of the main focus is the yeast and the molds. Yeast, asexual, and then molds are multicellular, and they are sexually produced. So that's just a quick review of fungi. Just pretty much know the PowerPoint. Bacteria. I've been asked on Piazza, the bacteria that she gives you on the study guide, is that a good thing to remember? Of course, I've said, Dr. Kokash was very good laying out the bacteria. Gram-positive cocci, gram-negative cocci, gram-positive rods, gram-negative rods, which has the most. I've always told the students that they have the most, so I would always start with the less amount and go to the most amount. So, like I said, gram-negative rods have the most, and then gram-negative spirals, and then spirochetes. Yes, she laid it out perfect for you guys. She did a very fantastic job. She did you guys a huge favor doing that. That is not all I would know from that. If you go to the PowerPoint for bacteria, it gives a lot of the characteristics. I would know the characteristics. It would just be nice to know the characteristics, because not only are you going to need this for this class and this exam, you're going to need it for the final exam, you're going to need it for, I'm sure, nursing exams, I'm sure you're going to need it for global health careers in the future, so it's good to know now. I would know the different characteristics, like Bacillus anthracis, which of course is anthrax. It's the disease of the cattle that was transmitted to humans, and now it can be used as a bioterrorist weapon. I would know microbacteria, which is gram-positive. It's acid-fast. It's slow growers. What's another big one? Lactobacillus is, of course, a gram-positive bacilli for milk and yogurts. It's found in the intestines and oral cavities of humans. It secretes an acid, which is used in industry to make yogurt, sauerkraut, and pickles. I would know Staphylococcus, of course, which is Staphylo aureus. It's the most important human pathogen. So, yes, Dr. Kokash did a great job in characterizing if it was gram-positive cocci, gram-positive rod. I would know characteristics of each one that she lays out. So, for gram-negative rods, she gave you all of them. Shigella, E. coli, Salmonella, Bordetella, Yersinia. Yersinia is Black Plague. I do not know if that's going to be in your exam. That's just one of those things that you remember when I took the class 5 semesters ago. When I'm your learning assistant, I'll remember it when I'm 55 years old, doing whatever I'm doing at the time. If I hear Yersinia, I'm going to remember the Black Plague. Rixetta, which is transmitted by insects. Chlamydia, which that's an important one. That is a very important one. Chlamydia. We all know that it's an STD. But what is unique about chlamydia? It's unique because it has a unique lifestyle and use elementary bodies within that life cycle. I have seen that question a lot. So that's what, as Dr. Kokash did give you some of those helpful tidbits on the study guide, I would know them all. Because it's helpful. It's going to help in your future, and it's going to definitely help for this exam, and it's definitely going to help for the final exam. So that is about it. Like I said, PowerPoints, I know there's a lot. I know even though there's only 4 PowerPoints, there's a bunch of slides to each. But it would be beneficial to know them now. I'm sure you guys have been studying a lot. You guys have been asking great questions on Piazza. I have gotten back to almost all of them. I will get back to the rest by tonight, I promise. Unfortunately, as a learning assistant, I wish time would stop for my classes, but my professors already told me, keep up. So I'm trying to keep up with my own work. I had a critical book review today that was really stressful because it was a 500-page book and we have 3 weeks to read it. So I'm done with that, so I'm making your video now. I'm answering your questions on Piazza. Please ask more. It's great. I think Piazza, not only am I giving answers, but your classmates as well are giving answers, and every single one has been correct so far. So if you're not on Piazza, please sign up. There's a link under the Learning Assistance content on Blackboard. If you have any questions at all from this video, please email me. Please put it on Piazza. Dr. Kokash is, of course, having her review session. Last time I posted this video, Dr. Kokash said she had a great attendance at her review session. I hope that some of those students watched this video and then went to review session. That is a great thing to have, is to have a professor that cares to do a review session. I have not had professors do this unless we were running really extremely fast through the class and we had a free period. So Dr. Kokash is doing an amazing job. So please go to a review session tomorrow. Please take questions that you have from this video. If you cannot make it, please email me or put it on Piazza. Study hard, and I will see you guys Tuesday.